Hello and welcome back to my channel. In this part of the video, we are going to discuss about the RNA interference. What is RNA interference? RNA interference is a RNA which is responsible for different types of processes that takes place post-transcriptionally. Some of the RNA interference like the siRNA, small interference mRNA, miRNA, microRNA are responsible for the gene silencing process. What is gene silencing? You can say that is, if there is any defective gene in our body, in order to prevent their activity or to stop their activity, this mRNA is used. Okay, you can say that these are the other types of mRNA. What are they, what is their process, and all we will be discussing in this part of the video. So let's begin. What is RNA interference? You can say it is a biological mechanism by which double-stranded RNA induce gene silencing by targeting complementary mRNA for degradation. Degradation means to break down. Okay, what happens? A double-stranded RNA here refers to RNA interference, which will produce gene silencing to the target mRNA. Okay, it will target the mRNA which is needed to be silenced, its activity needed to be stopped down. So it will target that one and then it will stop their activity. And one of the important point is that gene silencing is done post transcriptionally, means after transcription, after the mRNA is formed, this process is done. If there is any defective mRNA, if there is any DNA which is needed to be stopped, its gene needed, its activity needed to be stopped, in that process this 2 mRNA is used. Okay, and why it is important? Because it helps in the regulation of certain gene, like if certain genes are responsible for causing any type of disease, or you can say if certain disease is attacked, if certain genes are attacked by a certain virus. So in order to stop or to silence that gene, to silence that cell, this process, that is gene silencing, comes a very important role. And as I have already told you that RNA interference is of two types, that is siRNA, small interference mRNA, and miRNA, that is microRNA. What is the process? What are they? We will be dealing with it each one in detail. So hope you have understood the basic what is gene silencing, why the RNA interference is used, and what is the target of it. Let's begin. First we will deal with the small interfering RNA. Okay, what is small interfering RNA? It is a double-stranded RNA. As you can see here, I have mentioned 3 points regarding the small interference RNA, and this diagram represents the mechanism how it works. Okay, so let's begin. Wait. Okay, so it is a small double-stranded RNA. Okay, and it is a non-coding RNA molecule. Non-coding means what? As I have already told you, that they do not take part in the protein synthesis process. Okay, and their length will be 20 to 25 base pair in length. So you can see here, this is the double-stranded RNA. Okay, long double-stranded RNA. This is the 5' end and this is the 3' end. What happens? An enzyme called dicer, you can see here this green color, an enzyme called dicer comes and bind to this double-stranded double-stranded RNA structure. After binding to it, what it will do? It will cleave the structure in this middle portion. It will cleave and it will make this portion into a short siRNA. Okay, first it was long double-stranded RNA, then dicer molecule or dicer enzyme will come and bind here and it will cleave. From here it will cleave here like this and it will remove this much part. So it will become a short RNA, or you can say small interfering RNA. Then what happens? As a result of this cleaving, there will be a overhanging region in both the side. There will be overhanging region in both the side. Then what happened? Another 2 protein, that is argonoid 2 protein, AGO2 means argonoid, and TRB protein. Okay, these are the 2 proteins which comes and bind to this that is small siRNA, and this leads to the formation of a risk factor, what is known as RSISC, that is a risk complex will be formed after the binding of this two protein. Okay, a complex will be formed, you can see here a complex will be formed here. Then what happens after that risk binding, then what will happen with this among this two strand, one strand will act as a passenger strand which will be cleaved out later on, another strand will be act as a guide strand. As the name suggests, the guide strand will act as a guidance or a mentor which will guide through the mRNA and will reach out the target mRNA which is needed to be cleaved out, and it will bind and form a complementary pairing with that mRNA which is needed to be cleaved. That's why that is the role of this guide RNAs, guide strand. Okay, so this guide strand will guide the mRNA which target, which will bind to the target mRNA which is needed to be cleaved out. So what will happen after this risk loading all is done, the guide strand selection will be done. Here the guide strand selection, and passenger strand, which is this strand, will be cleaved out or will be ejected out. So now only a single strand will remain, that is known as the guide strand. Now what will do, the guide strand will move along the mRNA and it will reach to the point where the mRNA needed to be cleaved out. So you can see this is the target mRNA, that is 5 prime to 3 prime, and it will recognize and will form complementary base pair with it. After doing so, it will cleave the strand where it is needed to be, and then after that the recycle of the cleaved mRNA and risk recycling. After that what will happen, this guide strand and the protein which was binding to this mRNA will be released out and it will be recycled. What do you mean by recycle? Again it will be reused for the another cycle to be complex, and now the gene which is needed to be cleaved out or needed to be silenced is removed. Okay, so this is the overall process which takes place in the small interference RNA. If you understand this one, this is the generalized process. If you understand this process, then the next process, that is the microRNA, will be very easy. Hope you understood this. If you have any doubt, you can ask me. Next, moving to the microRNA. MicroRNA is a small non-coding mRNA molecule. What is the major difference between the small interference RNA and this microRNA? The similarity is that both are non-coding. But what is the difference? Difference is that small interfering RNA is a double-stranded RNA. Okay, whereas microRNA is a single-stranded RNA. This is the difference. Okay, and microRNA is found in plants, okay, animals and some viruses, and the function in the RNA silencing. So it is found everywhere and it will help in the process of RNA silencing or gene silencing. They function by base pairing with the complementary sequence with the mRNA molecule as the single-stranded or small siRNA was doing. They first present in the nucleus, then moves to the cytoplasm. This is the second difference between the siRNA and microRNA. First difference is that siRNA is double-stranded, whereas second difference then microRNA is single-stranded. Second difference is that that is the siRNA is present in the cytoplasm itself, whereas microRNA will be first present in the nucleus, then it will move to the cytoplasm. So these two main difference will be the difference this needed to be understood clearly. Okay, if it is asked in the exam or in an interview or in a viva voice, you can easily differentiate it with. And what is the similarity? Similarity is that they are both involved in the gene silencing process and they are the non-coding RNA molecule. That is, they do not take part in the protein synthesis, rather they are involved in the RNA silencing. Okay, so let's begin with this process. So what happens? This is the host RNA, or you can say the host gene, which is needed to be cleaved out. Okay, then what happens? Second, that is pre-mRNA or similar, that is pri-mRNA molecule is formed. Okay, what will happen? What is known as pri-mRNA? Pri-mRNA means that is it has 5 prime cap and the polyadenylation tail, and it is the pri-mRNA, means the native mRNA which is formed immediately after the translation process. This is needed to be processed out so that it will form the pre-miRNA. The process you can see here, that is the 5 prime cap, and this polyadenylation is simplified and it is shortened down. Okay, you can see here, that is here the pri-miRNA, you can pronounce it as pri because this is pronounced as pre. So this pri-miRNA is shortened or it is cleaved out into shorter molecule, that is pre-miRNA, by two protein, you can say, that is drosha and pasha. Okay, so what does they do? They, by the help of spliceosome, they will cleave out this region, extra region, intron region, they will cleave out and they will make this longer chain into a shorter pre-miRNA. But this all takes place in the nucleus. This is very important point to be noted down. But we need to come out into the cytoplasm region. So you remember that this processing from pri-mRNA to pre-mRNA takes place in the nucleus because it is asked in the exam. And where these proteins are present, that is dosha and pasha, they are present in the nucleus. This is tricky questions which are asked in exam. So you need to be very careful. Then what happens, a protein that is known as exportin 5, that is a transporter which helps in the transport of this pre-miRNA into the cytoplasm region. Obviously with the help of GTP it will come into the nucleus, cytoplasm. After coming to the cytoplasm, the process of what takes place in the siRNA, same process takes place here. That is, dicer will come, you can see dicer will come and cleave into shorter, that is miRNA, it will shorter, and then two strands will be formed. One is passenger, one is guide strand. Guide strand will be removed, passenger strand will be removed out. Obviously AGO2 and that TRPB, that is argonoid and TRPB protein, both will bind, then they will form the passenger strand and the guide strand. Passenger strand will be cleaved out, guide strand will be present, and then it will bind to the targeted region, and then the same process up till this region there is little bit difference. After it comes to the cytoplasm, the process is same that takes place in the small interference RNA. That's why I told you, if you understood this mechanism clearly, you can easily get through this by understanding only certain things. That is, the first part is important, that is also very simple, then rest of the thing is same. So these are the two process mechanism which takes place in the gene silencing process. Then this is a table which I have summarized it, that is mainly is for your better convincing or exam, you can easily go through this. miRNA is known as microRNA. It is present in animal, plants, protist. Okay. Sometimes it is asked which type of RNA which is responsible for gene silencing in protist is present. That is, which type of RNA, miRNA. So you can see here from examples also they will cross question the make a question. That's why I am given this example. Animals, plants and protists, this miRNA is present, and they have dicer as you have seen the process, and drosha dependent. What is drosha? Drosha is a protein which is found in the nucleus. Okay. Why it is required? It is required for the conversion of pri-mRNA into pre-mRNA. Next, coming to the small interference RNA, it is animal, then fungi, plants and protists, they are present, and dicer independent. Sorry, I have written here dependent, but they are independent because you have seen in this process no that is drosha is required for this process. Okay. And piRNA, the metajoins it is present, and dicer. These two, obviously these two are very important for the exam point of view. So this is all about the RNA interference in detail, and with this we come to the end of the molecular biology series, and hope you understood this. There are certain other chapters also in their molecular biology, but I think we need to give a little break to this molecular biology series We will come again if there is any other video. There are so many molecular biology topic, but this series is been really lengthy. That's why I want to switch some other series, or I may come with another molecular biology, but I want to try something new series so that you will find it interesting. And if you have not subscribe to my channel, to subscribe it, like and share among your friends, because these are very very basic things which are oftenly asked in exam. And that's all for today, and have a nice day. Thank you.